Good afternoon, everyone. My name is Windy, founder and CEO of Resistomap. We provide antibiotic resistant monitoring service and we are based in Helsinki, Finland. I'm very happy to share with you to celebrate World Antimicrobial Resistance Awareness Week in 2023, and I will discuss about our activities in Resistomap for the global monitoring of antimicrobial resistance in the environment as a One Health approach. But first of all, before we start about discuss about the antibiotic resistant monitoring, I would like to refresh our background that why bacteria becoming resistant to antibiotics. So there are two kind of primary pathway where this bacteria become resistant to antibiotics. First, by producing protein that can pump out the antibiotics inside the cell to out of the cell, or to decrease the uptake of these antibiotics inside the cell. Another pathway is by modifying the target that antibiotics affect. For example, by produce enzyme that can inactive antibiotics or can alter the target of antibiotics. And all of these activities are expressed from the antibiotic resistant genes. And that's why we can usually monitor the level of antibiotic resistance in bacteria by monitoring the genes of these antibiotic resistant genes in bacteria. Another information that why antibiotic resistant genes mobility is important to be monitored, because with this presence of these mobile genetic elements, bacteria or the genes can be transferred between bacteria or different kind of species in the community. So that's why it's very important to study also the mobility of these resistant genes to understand the spread of these resistant bacteria. Based on the study, these resistant genes, especially where it is a human-associated and clinically relevant bacteria, they are mobilized more than in other type of ecosystem. So this is very important of monitoring resistant bacteria, especially that are related to human or clinically relevant bacteria. And as we all know, of course, antibiotics is not used only in human or in the medicine, but also in agriculture, including in animal, in plant, and in aquaculture. All of these, and of course, in the pharmaceutical manufacture where antibiotics are produced. So when there are antibiotics, including when it's used by human or in animal, resistant bacteria will develop. And both antibiotics excretion, also resistant bacteria, and their resistant genes will release to the environment through our waste. For example, in the wastewater, where all the waste from the hospital and the community will release, or through the animal, where the manure are applied as a soil fertilizer. All of them will come back through our body or our microbiome through the water, directly interact with the environment or through the food chain system. So this is very important. We understand the spread of antibiotic resistance not only in the clinical setting or in the agriculture, in animal health setting, but also in the environment as a One Health approach. And that's why, as a cornerstone of the global strategy against antibiotic resistance, is to identify, characterize, and monitor the level of antibiotic resistance in hotspot where resistance is developing and spreading using One Health approach. How I end up become working on this antibiotic resistant monitoring in environment. So it's starting when my PhD at the University of Helsinki. I learned that antibiotic resistance is not only a problem in the hospital, but also in animal farm. My PhD focusing on antibiotic resistance in aquaculture setting. And then after my PhD, I continue as a postdoctoral researcher where I want to bring all this technology to my home country in Indonesia. I'm originally from Indonesia, yes. And I want to understand the level of this antibiotic resistance, especially in the water ecosystem. So we study from the spring water, where is the source of the water, and then follow the stream of the river, whether it get infected from agricultural, in the rural area, from the hospital, also from the population in the urban area, uh, and then up to the estuary where the river connected to the Indian Ocean. The problem in Indonesia, or I believe in most on region where the lack of sanitation, wastewater from the urban setting or from the agriculture, including the hospital, are released directly to the environment without or with lack of the proper wastewater treatment process. So this is one of the reason why uh, if we monitor the level of this resistant bacteria or resistant genes in the environment, we can identify the source of this resistant bacteria, especially from the wastewater that are not treated well. And based on this study, for example, in Indonesia, I have the urge of the need of monitoring of this antibiotic resistance also in the environment, because most people don't aware about this issue because it's not easy, right, to monitor antibiotic resistance in the environmental samples. And that's why one of the reason why I moved from academia to the entrepreneurship, to solve, or in this case, like how we can solve this issue to make sure that everyone have the access to this technology and knowledge on how to monitor antibiotic resistant pollutions in the environment. And this is why the problem that we would like to address is the access to the state of the art technology, because monitoring antibiotic resistance in environment require a molecular genetic method, since we study the genes of the resistant bacteria. And another problem is the data analysis, because when we talk about antibiotic resistance, there are hundreds or thousands of different kind of antibiotic resistant genes which convert to different kind of antibiotics group, right? Because there are already 10 major group of antibiotics. And when we talk about large data set, we need to know about the method standardization so that the data that we analyze are comparable. And another issue is there's a lack of expert availability, because when we talk about antibiotic resistant in environment, it's not only uh, from the clinical or from the veterinarian setting. We need also environmental microbiology methodology in this process, and that's why we need more expert, not only AMR in the clinical animal setting but also in the environmental settings. And that's why Resistomap was born. We provide an end-to-end service to monitor these resistant genes, the mobile genetic elements, and also the pathogens from the environmental samples, faster and cost efficient. So we provide currently mainly for the research sector, where our customer are coming from different universities, research institute, also the governmental agencies. But we also provide a service to private sector, for example, for the water utility, for the wastewater treatment plant, or from the hospital for the wastewater monitoring, and then pharma company from the antibiotic manufacturers, including from agriculture and food sector for the food safety. And this is the number of projects that has been we have been served since 2019. So since we have been around for 5 years, and we have served over 350 projects from 120 institutions globally. And we analyze over 10,000 environmental samples across 45 countries. As you can see here, that this is the number of project. Mainly, we have a project coming from the UK or from Finland. And this is the type of environmental samples that we have analyzed. As you can see here, that the variation of the samples are really broad, including from the wildlife and also from the agricultural and from the reservoir or from the natural environment. And this is how it works. We usually deliver the result within 10 working days. So we communicate how to prepare the samples, and then we will organize to pick up the sample wherever you are in the world, including from Mexico. And as long as you have a DHL service, and that when the sample arrive in our laboratory in Helsinki, we will analyze, and we have data quality control, making sure the data produced from the laboratory are high quality, and we deliver the result in an interactive platform so that you can access this result wherever you are as long as you have the internet access, and you can share also with your colleagues through this interactive dashboard. And this is the type of environmental samples that we can analyze. We use environmental DNA method. So we isolate the environmental DNA from this all of environmental samples. Can be wastewater, surface water, manure, soil, including surface area. We analyze from this DNA, from the environmental DNA, we quantify the presence and the quantity of these genes in the sample. So we use high throughput qPCR called Smart Chip qPCR system from the Takara Bio, where they have over 5,000 reactions in one analysis. So if we compare this technology to standard qPCR, where there's 96 plates, this smart chip has meant that over 50 times more efficient. And we can run the analysis as the same amount of when we perform qPCR, which is around 4 hours. But then we can get the result 50 times more than standard qPCR. So that's why it's called the high throughput qPCR. And the nice thing on this system is also the flexibility to customize the configuration in one chip analysis. So you can analyze as much as up to 384 genes for 4 different samples, or for the wastewater, for example, mainly targeting 96 genes, you can analyze 18 samples in the chip, or you can target only as few as 12 genes for 128 sample. This usually used for targeting isolate bacteria, that if you want to know what pathogens of this isolate and what resistant genes carry in this isolate. So to give you better selection, which isolate that you should go for the whole genome sequencing. And then this is the gene selection that are available in our database. So you can choose different kind of up to 620 different kind of genes to monitor, include all the resistant bacteria, resistant genes to all 10 major group of antibiotics, for mobile genetic elements here, and also for pathogens. So there are around 20 pathogens, which are mainly for the clinical pathogens, a priority, and also from the water quality type of pathogens. This is the result. That way you can have a ResistUp interactive dashboard that you can see the number of detected genes grouped by the antibiotics, and then the relative abundance and the copy numbers of the genes measured, and also the heat map to see the profile of the result. And this is how the data flow from the Smart Chip qPCR system or any method. It goes to our database in the cloud, where we have the admin to manage how the data you want to produce, how the data you want to use. It can be for the public sector, or like from the university. If you want to publish the data, we will open the dashboard. But for the coming from private sector or from the very sensitive region, you might want to keep the data for only authorized person. So we can flexible to organize how the data been shared. And this is the new initiative that we are working on now, is the global monitoring of these resistant genes and mobile genetic element in urban wastewater. So from these over 10,000 environmental samples, there are some countries that we analyze from the urban wastewater. As you can see here, unfortunately, the level of resistance are pretty high. So this is the median of the relative abundance of these genes in proportion to 16S RNA gene, which is considered also the number of bacteria. So the red color means it's usually when they are reaching around 1 in 1,000 bacteria, the genes presence. So it's mean that the proportion of the genes in the total bacterial community. When you can detect 1 in around 1,000 bacteria, is considered as a pollution or beyond the natural level of resistant bacteria in the environment. And now this is like the whole type of antibiotics, resistant to whole group of antibiotics. But interestingly here, that when you focus on certain group of antibiotics, let's say to sulfonamide is the first antibiotics that manufactured by human in 1930s. So as you can see here that all urban wastewater means that it's reflecting our gut microbiomes, right? All type, all the resistant genes encoding resistant bacteria are abundant or means that are already high in urban wastewater. And this is the reason why also sulfonamide is not any more efficient to treat any infectious in human disease. And then if we go to carbapenem, which is, I believe everyone knows, that is one of the last resource of antibiotics that are currently mainly used to as a treatment for infectious, bacterial infectious treatment. As you can see here that uh, certain regions are really high. I think it's also depending on the amount of antibiotic use. But then if we focus only on the class A of carbapenem, it seems like in the south part of the world has much more resistant genes level. And then if we go to the next one, the class D, it seems like go more to the north side. So this is the reason why global monitoring is important, because the problem of carbapenem, for example, uh, carbapenem class A is mainly in the kind of in Asia, not necessarily becoming a problem in Europe. But then for the class D, it seems like there are only regions has potentially issue on this resistant genes to the class D. So this is the important why we need to monitor globally to all type of antibiotic resistance to understand better what is the current situation of antibiotic resistant level in the community level. And to conclude, with routine wastewater monitoring, the data can be used to measure the risk of resistant prevalence and spread, and as an early warning for potential outbreaks of resistant bacteria. Very happy to be here again. And any question or any information that you would like to know more, feel free to contact me by email or by WhatsApp also. And greeting from Helsinki. Thank you.